I'm going to be talking about bees. Is it okay if I don't use a microphone? Can everybody hear me? Great. I like to walk around. Okay, well, some background information about bees. So I want to remind people that bees are defined as insects that depend 100% on flowers for their sustenance. They get two main types of food. One of them is nectar, which is a source of sugar and water for the adults, and the females also collect the nectar, which they take to their nest, and they mix with pollen to form the larval provisions. The second main food source is the main topic of my talk, and that is pollen. Pollen is the exclusive source of proteins, essential lipids, vitamins, and minerals for the bees. And the pollen is collected by the female bees and carried in pollen baskets in the case of social bees, or in the scopal hairs for solitary bees, which may be on the legs or on the underside of the abdomens. The pollen is then carried back to the nest, where it is mixed with various amounts of nectar to form the pollen ball for the larvae. However, another aspect of pollen consumption, which has not been addressed very much in the literature, is that pollen is also a food for the adult bees. And so together with a colleague, Jim Kane, as well as subsequently many of my undergraduate students, we've been showing that adult females of several solitary bees consume pollen. So we've looked at four different females of four different species that you see up there, and we have found in each case that over 90% of the females that we have dissected, so we dissect each bee, quite carefully, as you can see here, and remove the digestive tract and score for pollen presence. So over 90% of the females had pollen in the digestive tract, and the females were shown to eat pollen throughout the flight season. We've also looked at males of several species, and we found that they too eat pollen, but in much smaller quantities, and they tend to eat it primarily right after they emerge from the nest, which is when mating occurs. So given that bees, as adults, intentionally, that is actively and with intention, consume pollen, the question arises, are the adult bees selective in the flowers that they use for pollen consumption? And more specifically, do females consume the same pollen that they collect for their nest provisions? So I should mention also, before I delve into this, that bees show different selectivity in terms of the flowers that they use to collect pollen, and are generally divided into two main groups. Elite-elected bees are a species where all the females restrict their pollen collecting to only a few closely related plant species, usually at the plant genus level, sometimes more broadly at the plant family level. And poly-elected bees are those then where the females will visit a variety of plants that are unrelated as sources of pollen. So the question becomes then, more specifically, is the composition of the pollen in the gut the same as in the scopy? So the general methods for all of our bees is we collect the bees. Typically we like to collect them at the nest sites if we know where those nest sites are. Otherwise we collect them at the flowers. We kill the bees by freezing. We then prepare pollen samples from both the scopal hairs and from the gut. And in all cases we mount the pollen on microscope sides in glycerin jelly stained with fuchsia red. For collecting pollen from the scopal hairs, we remove pollen from different layers within the scopy, so we can get pollen that was collected at different times during a single foraging trip. And for the case of the gut, then we laboriously dissect bees, removing one tergite at a time, and some of these bees are very small, only 1 centimeter long, and then expose the digestive tract, which is then removed without puncturing, or that the data for that bee are lost. And insects have a digestive tract, which has three major regions: the foregut, mostly the crop, the midgut, where most of the digestion occurs, and the hindgut. And you can see the Malpighian tubules, which are essentially kidneys in the insects, which separate the midgut from the hindgut. And the hindgut is further divided into the ileum here, and then into the rectum. And so we took pollen from wherever the pollen was present, but mostly from the midgut. Once we made these pollen slides of the scopal pollen and of the gut, then we scanned the slides to count up to 100 grains at least, And for each pollen type, we also took measurements of the size so that we can convert the percent composition to volume and not just use numbers of pollen grains. And we identified the pollen grains by comparing them with reference collections that we collected from plants that were blooming in the surrounding of the nesting site. So here you can see an example of pollen from a midgut of one bee, and here is another. And here you can see some reference pollen, and also the scope of pollen would look very much like that as well. So we've looked at both oligolectic bees and at polylectic bees. So in terms of oligolectic bees, we've looked at 4 different species. We looked at 3 that are nearly oligolectic, and one of them is oligolectic to buttercups, and this is in Sweden, and they're collected at the nest site. They nest in these thatch roofs, and the stems are phragmites. And then we have 2 species that are oligolectic on campanula, and these were collected at the flowers. We've also looked at one broadly oligolectic bee, Heidi Dobson, which is a specialist on the family Asteraceae, and most of the bees were collected at trap nests that we set out. And so here's the data for the 3 nearly oligolectic bees. I'm only showing the data for what we found in the gut, because the pollen composition of the scopy is very well established as being pure host pollen. And we can see that for each of these 3 species, here we have mean percent of pollen grains per specimen, and we see that actually a very high percentage of the pollen for each of the 3 species. In fact, over 90% of the pollen in each of these bees was the host pollen. And so we see then that the bees are showing the same degree of flower specialization for collecting pollen as they do for feeding on pollen. This is similar to some findings that were found by Shepila in 1978 in Finland, where he actually took museum specimens of some bees that were oligolectic, and he examined the pollen in these dried bee specimens, and he found that the pollen in the gut that he was able to find was the same as that in the scopal hairs. In the more broadly oligolectic bee that we looked at, Heroides truncorum, here then we're showing both the pollen in the gut, which is in these bars, and in the scopy here on the right. And so we found that all Asteraceae, as expected. We found that the bees had the same pollen in the gut as they had in the scopy, but there was some discrepancies in terms of the representation of some. So we can see the unknown here is a little bit higher in the scopa, whereas Hypocaris was a little bit higher in the gut. And some of the reasons for why there might be some of these discrepancies might be that some pollen is harder for the bees to pack into the scopal hairs, depending on the size of the grains and depending on how spiny they are. That might make it more difficult for the bee to pack it into their hairs. So overall then, we're finding even in this broadly oligolectic bee that they are eating the same pollen that they are collecting for the larvae. Well, we've looked at one polylectic bee, and this is Halictus rubicundus. So the Halictidae, the small sweat bees, are kind of our weedy bees today. As our bee diversity decreases, you can always find Halictus out there. And Halictus rubicundus is, like a lot of the Halictus species, are subsocial, so they may have multiple females in a single nest, and there may be actually more than one generation. So we actually collected the bees at the nest site. At the nest sites, which are actually at the Whitman College campus, right near the railroad tracks. You can see all the holes here. You can see a female just coming out of her nest. And we have 2 years of data. One of them is 2016, where we got the first part of the flight season, and 2017 we got the latter part of the flight season. And our reference slides were mostly collected from plants at a native plant garden that we have on our campus, which has a variety of different plant species. But there are also quite a few plants on the Whitman campus, and so we're actually still in the process of identifying all the pollen grains, but we have separated the different types of grains in our samples. Okay, so here are the data for the scopalodes. So what we have here, 2016 with 15 bees and 2017. Under here are the individual bees, so this is the identification number that they were given. And then we have the dates when the bees were collected. And over here we have the percent composition of the scopolodes in terms of volume. And if we look at these bars, and I should mention before looking at the colors, that these are colors for the 2016 species, and these are colors for the 2017 colors, species of pollen. And in each year, the bees were collecting pollen from about 9 different plant species. We found that if you progress here, you'll see that some colors disappear and new colors appear as the season moves on. So the bees are being polylectic. They are moving to new plant species as others bloom out. So they are following, as expected, the phenology of the plants. We found that individual females carry 1 type of pollen, 2 types of pollen, or even sometimes they carried 3 and possibly even more. But mostly, we found that 73% of the bees in 2016 and 67% of the bees in 2017 had pure pollen notes. So this is quite common in polylectic bees, that even though as a species the bees visit a variety of different plants for their pollen, individual females during single foraging trips might collect pollen from only a single species. All right, so now what happens in terms of the gut? So on our next slide then, so this is what we just saw above here, okay? And now these are the gut contents for each of those bees. And if you look in general, and you just compare the bars in each case, you will see that the gut contains a lot more colors for each bee than does the scopa. So the bees in general were eating a lot more different pollens than they were collecting. We found that of the 21 bees that had pure scopolodes and up here, we found that only 7 of them had also pure loads in their gut. And those are indicated by these lines here. And of those, 5 of them had the same pollen in the gut as in the scopy, but 2 of them had completely different pollen in the gut than the scopy. Of all the other bees, we find that then typically the gut has a lot more pollen than does, different kinds of pollen than does the scopy. There were some exceptions. You can look at these ones with the dark circles, where there are 2 kinds here and 2 kinds here, but maybe different combinations of pollen. You can also see some of the increase in diversity in some of these, in the light-colored ones. I circled some of the gray ones, where if you compare, they had pure scopy, but sometimes a really high diversity of different pollen in their gut. So clearly then the bees are feeding on a lot of different more plants than they are actually collecting pollen on. Also, if we look at some of the gut compositions, we see some of these pollen grains are present in less than 10% of the composition, and those are probably more contaminations, possibly picked up by the bees as they're feeding on nectar. Then they're also ingesting some of the pollen at the same time. So it suggests it's not so much intentional feeding as much as we saw earlier. I should mention that these results are similar to a study that just came out by Russell and Danforth, where they actually looked at the gut and the scopopollen of a particular Serotina species, and they found actually that the guts were more diverse as well than the scopolodes. Okay, so in summary then, we found that the electric species then were as specialized in the pollen feeding as in the pollen collecting. And this suggests that the term oligolecty, which refers to pollen collecting, should be applied to adult pollen feeding as well. But before doing that, we should collect more data to confirm this generalization. In terms of polylectic species, we see that the adult females foraged on a greater diversity, a great diversity of plants in general, but they had a higher frequency of constancy in pollen collecting, so a higher diversity of plants were used as sources of pollen for consumption. So what is the source of this discrepancy between pollen foraging in terms of collecting and pollen foraging in terms of feeding? And 2 possibilities. One, I mentioned earlier, that possibly the bees have more difficulty packing certain pollen into the scopa hairs. Bees vary in terms of the density of the scopa hairs. And there might be actually other sensory stimuli that are involved in these 2 behaviors. And also many thanks to my undergraduate students and to funding sources. Thank you.